Hello. Welcome to the next in a series of review clips designed to assist you with your Biology 3201 public examination preparation. In this clip, we'll be taking a close look at DNA, certainly examining DNA as being the code of life. We now know that DNA, the molecule, forms a molecular basis on which inheritance occurs. The information necessary to sustain and perpetuate life is found within this molecule. This is the genetic material that is passed on from one generation to the next. DNA forms a blueprint for building living organisms. And while we now accept that as being factual, this was not always the case. It was unthinkable in the early 1800s for the leading scientists and philosophers of the day that a simple chemical molecule could possibly hold enough information to build a human being or any other organism. They firmly believed and was entrenched that all plants, all animals, all life had been specifically designed by a creator. There were a number of scientists involved in this process. One of the first was Friedrich Miescher. What Miescher did was he isolated a substance that he termed nuclein from white blood cells. He realized that nuclein was composed of an acid portion that he entitled nucleic acids and an alkaline portion that was later shown to be a protein. It was called nucleic acid because he isolated it from the nucleus. Post-Miescher research showed that nuclein was made of the series of strand-like complex of nucleic acids and proteins that were tightly bound together. We now know these as chromosomes. Next came along Levine, Levine. Levine isolated two types of nucleic acid that can be distinguished by the type of sugar they contain. RNA contains ribose. DNA contains deoxyribose. He showed that these nucleic acids were made up of long chains of individual units called nucleotides. Nucleotides are comprised of a 5-carbon sugar, a phosphate group, and a nitrogenous base. There are 4 different bases in each nucleic acid. Nitrogen bases contained in DNA are as follows: adenine, symbolized A, guanine, symbolized G, and cytosine, symbolized C. The 4th in DNA is thymine, or T. In RNA, ribonucleic acid, the bases are adenine, guanine, and cytosine, but there is no thymine. Uracil replaces thymine in RNA. Levine felt that DNA and RNA contained equal amounts of nucleotides. This was something that was an error, and he was later disproved. Friedrich Griffith, moved along the idea of DNA as being a hereditary molecule, and he studied bacteria and pneumonia. What Griffith found was that if you took dead pathogenic bacteria, they could pass on their properties to live non-pathogenic bacteria. These bacteria were transformed, and he called this ability a transforming principle. The transforming principle is that genetic information can be transferred from one organism to another. So what Griffith did in 1928 was he took living S strain bacteria. Pneumonia comes in 2 strains, S and R. The S strain is lethal. If you put S bacteria in a mouse, it dies. R strain bacteria has no impact on a mouse. The mouse would live, even if injected with R strain bacteria. Yet, if he killed S strain, the mouse would die. It lived, rather. Yet, if the bacteria was killed and mixed with live R strain, and you injected that concoction into a mouse, the mouse itself would die. And by analyzing the dead mouse tissues, you found that there were both live S and R strain bacteria. Either the S bacteria came back to life, which we know wouldn't have happened, or the R strain were somehow converted, transformed into S. This was indeed the case. To try and further cement Griffith's work and Griffith's transforming principle, Avery, MacLeod, and McCarty did further work. What they did is they treated bacteria with protein-destroying enzyme, and yet the transformation still occurred if you destroy the protein. If you destroy DNA, no transformation. If you treat a bacteria with RNA, but not DNA, the transformation still occurred. Only by eliminating DNA would transformation be stopped. Thus, by the process of elimination, Avery, MacLeod, and McCarty proved that DNA was the material responsible for the transforming principle. Chargaff came up with some important ideas as well as it pertained to DNA. Chargaff overturned Levine's conclusion about nucleotides. He found that the 4 nucleotides were not present in equal amounts, but rather in varying proportions. He found that the amount of nucleotides in DNA, A, C, T, and G, does indeed vary from species to species, and the nucleotide composition for members of the same species is constant. And in fact, in any sample of DNA, the amount of adenine equals thymine, and guanine equals cytosine. This is known as Chargaff's rules. And Chargaff's rule is, in any sample of DNA, the amount of adenine equals thymine, and the amount of guanine equals cytosine. A equals T, G equals C. Hershey and Chase also made some groundbreaking work as it pertained to certifying that DNA was indeed the hereditary molecule. In fact, Hershey and Chase gave the final proof that DNA and not proteins carry genetic information. They did their work using radioactive labeling in an experiment with a virus called a bacteriophage. Simply put, a bacteriophage is a virus that infects a bacterial cell. What they did was they ran 2 experiments at the same time, both on bacteriophages. They added a radioactive marker to the DNA of one phage. They added a radioactive marker to the protein of another. They allowed the phages to infect bacteria, E. coli to be specific. The 2 samples of bacteria were observed, and the first sample, which was exposed to radioactive DNA, the bacteria became radioactive, but the fluid outside did not. In the second sample, which had the radioactive protein coat, the bacteria did not become radioactive, but the surrounding fluid did. Their conclusion, based on these results, was that only the DNA from the virus entered the bacterial cells. Therefore, transmission of material from the virus to bacteria happened only because DNA was injected. Thus, as a result of this work, DNA became known as the molecule that transmits genetic information. So to summarize the Hershey-Chase experiment, they tagged radioactive sulfur in phages, they broke it apart, they lysed it, let the bacteria scope, and they showed no sulfur. Sulfur is part of protein. They did the same thing with phosphorus. And when they went through with phosphorus, they found that phosphorus-labeled DNA was actually showing up in the bacterial cells. So by having the radioactive phosphorus, this showed that because phosphorus is part of DNA, that it was indeed DNA that was being passed from virus to bacteria. So in summary, the Griffith experiment provided evidence for transformation. Hershey and Chase gave credence that it was DNA. Other evidence included the DNA volume doubles before cells divide, replication. Chargaff's ratio of nucleotides, A equaled T, G equals C, gave credence to DNA as being the hereditary molecule. Wilkinson Franklin in the 1930s did work further on determining the structure of nucleic acids and DNA. Their X-ray pictures proved that DNA had a helical, twisted shape, and they found that the DNA had 2 distinct and repeating patterns. The bases were hydrophobic and contained within the center. The sugar phosphate backbone was hydrophilic and was located on the outside. And in the 1950s, Watson and Crick took the work of others, and they worked on different models using the knowledge of Wilkinson Franklin and others, and they proposed a structural model of DNA that is still in use today, and they called their model the double helix. Past public examinations have had questions posed on this material. This question, as lifted directly from a past public examination, reads as follows. Who proposed the double helix model of DNA? Franklin and Wilkins, Hershey and Chase, Sutton and Boveri, Watson and Crick. The correct answer is indeed Watson and Crick. This question here, as drawn from a past public examination, asks you to utilize Chargaff's principles. And you're asked what percent of guanine will be found in a strand of DNA containing 28% cytosine. If you recall, C equals G. A equals T. The amount of cytosine is 28%. Because the amount of cytosine is equal to the amount of guanine, the correct answer must indeed be B, 28%. C equals G, A equals T. If a sample contains 28% cytosine, it must as well contain 28% guanine. And this question as posed asks you which researcher isolated two types of nucleic acids. Watson was responsible for the structure along with Crick. Mendel proposed Mendelian laws, the law of dominance, segregation, independent assortment. Barbara McClintock worked on jumping genes, transposons. The correct answer is A, Levine. Thank you for your patience. I hope you found this learning clip to be helpful.